So today's lesson, we're going to continue on from where we left off yesterday. All right. So yesterday's lesson, we looked at basically the first two steps of gene expression, which is transcription and RNA splicing. So transcription, we know it as being okay, let me talk about the gene first. Okay. So first thing we know about the genetic code, it's read 3 at a time. Second thing, it has certain properties. First, that it's universal. All living things use the same amino acids. Next thing is that it's unambiguous, meaning that a codon will always code for one amino acid, right? Any codon will always code for only one amino acid. Okay. Degenerative, meaning that it has redundancies that are built into it for accounting for mutation. Next is that it's non-overlapping, meaning that each codon is read one at a time rather than all together, right? Next, there are certain parts of a gene that you need to be well aware of. The most important ones are the operator, promoter, termination sequence, exons, and introns. Make sure you're aware that the operator is only present in the prokaryotic life, which are bacteria and other archaeas. Whereas with the promoter termination sequence, it's present in both prokaryote and eukaryote. With introns, these are the sequences that don't code for a protein, which you can find in the gene as well. They are only found in eukaryotic cells, not in prokaryotes. Prokaryotes are only having the exons when they are doing transcription. But with eukaryotic life, they also have introns within them that need to be filtered out during RNA splicing. So the next step, so transcription, easiest way to think about it is copying a page from a recipe book onto a blank piece of paper. You're making a pre-mRNA molecule based on the information on the DNA through transcription, right? So that works when the RNA polymerase binds to the gene region, the promoter region, and then starts the transcription from there, making an mRNA molecule, pre-mRNA molecule, until it reaches the termination sequence, at which point it detaches and the pre-mRNA will undergo RNA splicing. So pre-mRNA will then be made into mRNA by undergoing two changes. One is the addition of the methyl GCAP and the poly A tail. First thing you should know about the pre-mRNA is which direction is it made from. So it starts at the 5' and it's built up going towards the 3'. So the 5' is the front, 3' is the back. You can see here we have a pre-mRNA molecule. It has introns, it has exons, has no other molecules attached to it. So during RNA splice processing, a 5-methyl GCAP and a poly A tail will be attached to it, and also the introns will be taken out. Also, this is a chance for alternative splicing to happen, which is when different exons are either left out or joined together to make different proteins, right? All of these proteins are functionally viable, meaning that they are able to do their functions. It's just that they don't look the same, or they don't necessarily do the same function as well. They might be working on something else altogether. But the fact is, it was coded for using the same gene sequence. So only difference is that the exons that make up the protein are different. The video, I think we can skip. So today's lesson, we're going to look at translation, right? So we started with DNA, that is our storage, that is our library of information. Then we copied it into a blank piece of paper as transcribing. We will transcribe it, that's what we call transcription. Then now, once we've done RNA processing, which is to make it ready to be read, we are going to read it and translate it to make the food that we want, the protein that we want, okay? So when we think about how mRNA is made into a protein, there's a few steps that you need to take into consideration. First of all, this mRNA molecule has now come out of the nucleus, right? Remember that we started our journey in the nucleus. This is where the DNA is, and pre-mRNA is made. Then pre-mRNA is also processed inside the nucleus. Once that is done, the mRNA molecule will exit the nucleus and find the nearest ribosome. Okay, so for translation to happen, there are three RNA molecules that need to work together. So we would have looked at this when we looked at proteins as well, but these nucleic acids, but these RNAs are ribosomal RNA, transfer RNA, and messenger RNA. So the messenger RNA is the one that carries the information about the exons, the how to code for the protein. Transfer RNA is the RNA that brings the amino acids to the ribosome to be made into a long polypeptide chain. Ribosomal RNA is the RNA that makes up the ribosome. Okay, so when you think about the ribosome, you should be thinking of it as two subunits that make up one big ribosome. So, yeah, first one. So let's see. All right. So now we have a picture of a mRNA, a ribosomal unit, and also the tRNA coming in. So how does this process start? This process actually should be up there already. Yeah. Okay. So before I get into the process of how, we must always make sure we remember that these amino acids are coded for using the degenerate table, right? Remember the table I showed you guys? This is how amino acids are coded for. So we have an mRNA molecule. That mRNA molecule is built up of base pairs, right? A, C, G, and U. Based on what codon is read, 3 letters at a time, it will code for a specific amino acid. And we can see that it needs to start with the AUG. No matter what protein it is, no matter what stage of the thing is, it needs to always start with a AUG. So whenever we look at an mRNA molecule, the first codon will always see, right? Let's say if this is a long line of mRNA, if I looked at all the base pairs, the first 3 base pairs will always be A, U, G, because that is the start codon. That is the codon that will trigger the first amino acid to be released, which is MET. MET will make the first codon, the first amino acid of the protein molecule, and then it will continue from there. So the process is actually very straightforward when you think about it, but we need to make sure we understand it very well, right? So first things first, a small subunit of ribosomal RNA attaches to the 5' end of the mRNA. Remember that the 5' mRNA is where the transcription started. It started at 5' and then built itself from the 3' end. So you could say that 5' end is the top. So the small ribosomal RNA attaches itself to the top and then starts moving along until it reaches the start codon. Remember that the start codon is the signal for it to start translating, start making a protein molecule. Once it reaches the start codon, the tRNA molecule matches with that start codon. So the tRNA molecule is always carrying something called an anticodon, complementary anticodon, which is basically a way for it to match the right amino acid with the right base pairs. So let me show a picture. Should have put it together here. All right, so you can see here, right? For a tRNA molecule to work, for it to know which amino acid needs to go at the right place, it uses the mRNA molecule. So when it has an mRNA molecule, the tRNA has those bases associated with that that need to be complementary to that strand. So if it's not complementary, that means that amino acid is not supposed to be there. Only if it finds the complementary part to itself, it releases its amino acid and then gets released off. So let me try to put the process together. So imagine in the cytoplasm where the ribosome is located, there are a bunch of tRNA molecules. All of them are carrying one amino acid. So they're just free floating, they're moving randomly, right? So when one of them comes in contact with this ribosome, they will first test whether their anticodon matches with the mRNA's codon, whether it's complementary, right? In this example, we see CUA, we know that's complementary to G, A, and U. So once it finds a complementary strand, this triggers the amino acid to be released, linking together with the long chain of polypeptides. And then once it gets this position, it detaches from the amino acid and then gets released off at the third step. So this process continues all the way until the stop codon is reached at the 3' end of the molecule. So did I leave anything out here? Okay. So when we think first the tRNA molecule comes in, checks for the complementary antisense anticodon, and then once it finds the complementary anticodon, it releases the first amino acid, methionine. Methionine is the codon, the amino acid that you get for the start codon. So that's going to be the first amino acid of most proteins. It will always be methionine because it is the start codon. So it will always be the first amino acid that makes up a chain. Okay. Once that happens, the big subunit will complete the structure of the ribosome and continue the process of translation. So we start off with a small subunit, which is the bottom part, and then mRNA binds to it, finds the complementary anticodon. Only then that triggers the translation, causing the big subunit to bind together and promote translation. Okay, so once that's happening, the mRNA molecule will keep moving through the ribosome, and every codon will be read and matched with the complementary tRNA molecule. That will then cause another amino acid to join together with the chain, causing the chain to elongate as more and more join together. So remember that every amino acid is joined together using a simple bond known as a peptide bond, and that reaction is called a condensation polymerization reaction. Okay, see where am I? All right. So this process continually happens until we reach the stop codon. So once it reaches the stop codon, what happens is, if this is stop codon here, right? So this 1, 2, 3. So if this is the stop codon, once it reaches to that point, it reads the stop codon, but no tRNA molecule will, no amino acid will be released from the tRNA molecule. In fact, when the stop codon is read, all that happens is that translation is stops and the polypeptide chain is released. Simple as that. No more amino acids are joined together, even with the stop codon. It is purely just to stop the translation. Only with the start codon will an amino acid be joined together with the chain, but with the stop codon, there will be no amino acids that are associated with it. Yeah.
Happens.
This one?
Yeah.
All right. Mm-hmm. Alright, so I think in a better picture would be this one. So delete that one. This is have anything material in it. Alright, so chain. So imagine when this chain was starting out, right? There was only the small subunit, and then that mRNA is bind together with the small subunit, and then the first tRNA molecule finds that start codon. Okay, so the tRNA molecule binds to that start codon, sees whether it has a complementary strand to the start codon, and then if it does, the methionine then is created as the first of a long chain. Okay, that is the first step. And then the big subunit joins together to promote translation, and then the process starts there. So every time a codon is read, it moves 3 base pairs. Then the next codon, then it moves 3 pairs, then the next codon, right? So after methionine, the next codon is read, right? Let's say it is a random amino acid. Let's say it's LEU. Okay. So when LEU is read as a codon, what happens is a condensation reaction occurs between the amino acid on this molecule and the amino acid that is supposed to make up the chain. Once that reaction occurs and they've created a bond between each other, then the tRNA molecule moves one step. So essentially, a ribosome has three parts to it. There's A site, P site, and one more site, I forgot the name, but you don't have to remember that. That is a high level of biology that you learn in uni if you go to it. But essentially, there's three positions that tRNA can take, and at each position it is doing something else, right? At site A, it first tries to match itself with the complementary mRNA strand to see whether the amino acid is to be released. Once it finds the complementary strand, the amino acid is then bonded together with the polypeptide chain. Once it gets to the P side, it will then release the amino acid, allowing it to form a chain, and then the tRNA will be released from the ribosome altogether. So remember, the chain is only possible because of the reaction that occurs between amino acids, which is the condensation polymerization reaction, which allows the peptide bonds to form between the amino acids. This process continually happens, right? The mRNA is read three at a time, continually until it reaches the stop codon at the end. When it reaches the stop codon is read, and the ribosome gets a message saying translation's over. Just stop. Just release the polypeptide and release the mRNA. Your job is done. So, and that in turn allows the polypeptide to be released, and that is just the first step of a polypeptide then eventually being made into a protein. Okay, so this is how we get a polypeptide from a gene. It starts off with the transcription. Yeah, it starts off with transcription where it makes a free mRNA molecule, and then that free mRNA molecule is then processed through RNA processing to be made viable for translation, both in terms of stability and also in terms of generating a functional copy of a protein. And then the third step is the translation part, where the mRNA goes out of the nucleus, finds the ribosome, and then has tRNA joined together with a complementary anticodon to then build a polypeptide chain. Okay, any part of this still confusing? I think I'm going to show you guys a video. Actually, no, this one's pretty good. All right, this one's a pretty good summary of everything. So, whenever we have a need for a protein, your body sends a message to your cells, make that protein. Okay, so first things first, we need to get the instructions to make that protein. Basically, we need the recipe to make the food that we want. So we need to get that recipe instructions from DNA. DNA is located within the nucleus. So it's not meant to be moved. It's basically in cold storage, just staying there, only used for essential things, only used to get information rather than to carry out any task by itself. So first things first, a functional copy of that instructions need to be made, which is the RNA molecule. So the first step is to transcribe the gene from the DNA using RNA polymerase to make a pre-mRNA molecule using transcription. Then that pre-mRNA molecule is processed by adding a poly A tail to the 3' end and a methyl G cap to the 5' end to allow it to go outside of the nucleus. The introns inside the pre-mRNA molecule are also removed out, and sometimes alternative splicing can happen to remove some exons depending on the need of the cell. So once both of those are done, the pre-mRNA molecule becomes a mRNA molecule. Only when it becomes an mRNA molecule can it exit the nucleus and undergo the second stage, which is translation. Translation is started by the mRNA molecule binding to the small subunit of the ribosome, and then a tRNA molecule finding the mRNA. Sorry, actually it starts with the mRNA molecule binding with the small subunit, and then the small subunit reading the mRNA molecule until it gets to the start codon. When it gets to the start codon, a tRNA molecule will happen to bind to that small subunit of ribosome and see whether its anti-codon matches with the mRNA's codon. If it's complementary, that means it will start translation. The only way it becomes complementary is if the tRNA molecule is carrying methionine, because that is the start codon. Basically, the anti-codon for the unnecessarily confusing it. All right. So tRNA binds to the mRNA, binds to the ribosomal small ribosomal unit, finds the complementary strand. Remember that for translation to start, it needs to start with the AUG, the start codon. Okay, so the mRNA would have read, the small ribosomal unit would have read the mRNA molecule until it got to the start codon. Knowing that this is where translation is going to start, it will hold it there until the right tRNA molecule binds to that region. Okay, when the right tRNA binds, that carries the anti-codon for the start codon, which is what? UAC. So a tRNA molecule that has this anti-codon will have to bind to the start codon. Only when a tRNA molecule with this anti-codon binds to the the subunit of ribosome will translation be initiated. So translation is initiated by that tRNA molecule releasing methionine, and then that will continue. The big subunit of ribosome will bind together, which allows for translation to continue happening every codon. And at every codon, the tRNA molecule finds its complementary strand, releases the amino acid, and the amino acid binds to the first amino acid, forming a chain. And this process continues and continues until eventually the ribosome reaches the stop codon of the mRNA, triggering the ribosome to release the polypeptide chain and also to release the mRNA to indicate that the translation has finished. So it's a simple process once you break it down like that, but every step is important for it to finally make a polypeptide chain, which will then undergo folding and so on to become a functional protein. Any questions about this so far? Yeah, this is jokes from Miss Luan's slides. Even I don't get it. This one, sure. That one, I don't get. I have an RNA joke, but it might get lost in translation. But here's the thing, RNA doesn't get lost in translation. It's just used in translation. All right, what do you guys think? Yes. Yeah, obviously, yeah. Transcription. The first step is of gene expression is transcription of DNA. Then what's the next step? Can I get Ayman? What's the step after transcription? What is the name of the step that has I'm not going to say that. That has the introns being removed and also the metal cap and the poly A tail being added on. RNA what? There's another word after that. Yeah, RNA processing. Okay, it's transcription of DNA, RNA processing, and then translation. These three steps need to happen concurrently for a functional protein to emerge. So we've looked at all these genes and stuff, right? So you guys are aware of your chromosomes. So for you to have a sense of how small genes are, if these are your chromosomes and your chromosomes are already very small by themselves, one gene would be one band. Basically a band. That's how tightly coiled the DNA is. The fact that this one band has